All right, today we're going to be going over chapter 5, which is talking about the extension and modifications of basic principles. And basic principles referring to those from Mendelian principles. Up until this point, we've been talking about the foundation of genetics and those basic foundational principles of transmission genetics or classical genetics that were really cultivated on the foundation of Gregor Mendel's work with and study with pea plants, understanding the law of dominance, the law of segregation, and how when we have homozygous versus heterozygous, what that looks like, okay? Understanding the association with genotypes and phenotypes. But there are instances where there are exceptions to those rules, and we're going to go over some of them today during this lecture. What you're looking at here is a great hallmark example of an extension of or modification to those basic principles. And there's a slew of others that we're going to talk about, but this one in particular, which looks at the coiling direction of snails. There's the shells specifically of snails. And this whole concept that they found, and we'll go into this further in the lecture, but they found that the direction of shell coiling in the snails was not determined by what we would normally think of as the genotype based off of the normal Mendelian principles, but it was actually determined by what we call the genetic maternal effect. And we're going to go into that. But I will tell you that this effect defies or is an extension, not necessarily defies, but is an extension of Mendelian principles, as it does not follow the normal Mendelian ratios and principles that you are familiar with. So when we talk about these extensions or modifications to these basic principles, it's on the foundation of Mendel, okay? One in particular is understanding the law of dominance, which is one of, goes back to some of the principal foundations of Gregor Mendel. Okay. Mendel observed that dominance in all traits, he observed dominance in all traits that he studied extensively. But he was actually aware that not all characteristics exhibit dominance. Okay. An example of that is when he conducted crosses looking at the length of time that pea plants take to flowers, and he saw that there were some deviations from what he normally would view or identify. So what happens when you have genes at the same locus, or two versions of that gene, and each version of the same gene is identified as an allele, but it does not follow normal Mendelian principles? Okay, there are different types of dominance. Dominance can be understood in regard to how the phenotype of the heterozygote relates to the phenotype of the two homozygotes. Meaning, if you have a carrier, an instance where you have two different versions of a gene, okay, big A, little a, big R, little r. You compare that phenotype to the homozygous dominant phenotype and the homozygous recessive. It is a clear indication of if it deviates from the law of dominance. For an example, you'll see in this diagram here, let me activate this pin for you guys. You'll see here in this complete dominance example, which I'm going to explain to you what complete dominance is. You'll see that A1 genotype encodes for red flowers, okay? A2 codes for white flowers. But if the heterozygote is red, the A1 allele is going to be dominant over the A2 allele. And you can determine that by the phenotype. If the heterozygote is white, the A2 allele is dominant over the A1 allele, as you see here. Okay? So looking at the phenotype will help you understand, of the heterozygote will help you understand dominance. Okay? So in this case here, the complete dominance can be identified based off of looking at the heterozygote and comparing it to the two heterozygotes. So this would be complete dominance. Incomplete dominance is where you have, looking at the phenotype, if the phenotype of the heterozygote falls between the phenotypes of the two homozygotes, dominant is incomplete. That means neither allele is dominant over the other. So when they come together, you don't get the homozygous dominant phenotype, nor do you get the homozygous recessive phenotype. It provides, or what appears to be, an intermediate phenotype. Okay, that is incomplete dominance. Another example of that incomplete dominance we see here with the fruit color in the eggplant. It's inherited as an incomplete dominant trait. Most of the time when you have incomplete dominance, you're going to have, it defies the Mendelian ratios that you would expect to see. And in this case, when we have normal Mendelian principles, we have two alleles, one gene locus. Usually the basic ratios we would expect to see is a 3-to-1 ratio for phenotype. That is suggesting that even the carriers are exhibiting the dominant phenotype. So let's see if we can draw that out. Let's say we're going to use big R, little r, okay? Let's do a Punnett square to carry this out. Okay? Doing this Punnett square should render big R, little r. So here's our carrier or our heterozygote. We have another carrier or heterozygote here. And then here we have the homozygous recessive. Okay? So what we have is homozygous dominant, homozygous recessive, and then we have these two carriers or heterozygotes. Now, so the ratio for genotype will be 1 to 2 to 1. The phenotypic ratio is 3 to 1 because 1, 2, 3, based on the law of dominance, should still produce the dominant phenotype, and only 1 will produce the recessive phenotype. However, in incomplete dominance, that is not what we see. It deviates from this normal Mendelian ratio. What we see, as we see here for this eggplant, okay, they get a purple fruit color if it's homozygous dominant, and crossing it with a white eggplant is homozygous recessive. Allowing them to fertilize produces the F1 generation where they're all carriers, okay, and they have violet. So they produce almost like an intermediate color between purple and white. This is different from what we would expect to see from Mendelian principles, okay? Because we would expect to see under normal Mendelian principles that all of the fruit would be purple in F1 generation, like we see here. When you do an F2 generation, so you allow them to self-fertilize, okay, 2 carriers, you're going to get the F2 generation, progeny. And what we see here again defies the normal Mendelian principles, in that we would expect to see a 3-to-1 ratio, phenotypic ratio, 3 offspring exhibiting the dominant phenotype and 1 exhibiting the recessive phenotype. But what we are actually seeing in incomplete dominance is 3 different phenotypes: purple, violet, and white. And what this is showing is that neither of the alleles are producing enough to provide the potency, if I may say, of the homozygous dominant or the dominant trait. Okay, so it's not enough that one allele that is dominant, the capital P, right, in this case here, doesn't produce enough product to produce a normal dominant purple phenotype. So what you get is an intermediate form. This, again, deviates from the normal Mendelian principles. Okay, so this table here will help you, gives a definition of these terms that I've been talking about, but make sure you go back and read through and look at the experimentation to fully grasp that concept, right? That complete dominance, the phenotype of the heterozygote is the same as the phenotype of the homozygotes. That's complete dominance, okay? Incomplete dominance is where you have that intermediate that falls between the range of the 2 homozygotes, and that's the carrier. Codominance is where both alleles in a heterozygote are expressed. One is not more dominant over the other. So another component that I want to talk about when we're looking at phenotypes, okay, is the level of phenotype observed may affect dominance. What do you mean? Okay. The level referring to molecular versus physiological. An example of that is cystic fibrosis. Okay. Cystic fibrosis is it's an autosomal recessive disease, and it's caused from the mutation of the CFTR gene, which produces the receptor, that acts as a channel and allows or regulates chloride levels to pass in and out of the cell freely. And what that does is it reduces the amount of mucus that builds up on the outer portion of the cell. However, in individuals with cystic fibrosis, that channel is mutated, and so it stays closed and it prevents chloride ions from moving in and out. They build up on the inside of the cell, but the mucus builds up on the outside of the cell. Okay. Now, the issue with this specific component and why I talked about the levels, okay, is that most people have 2 copies of the normal CFTR allele and produce only functional CFTR protein, that channel protein. Those with cystic fibrosis produce 2 mutant CFTR alleles and produce only the defective CFTR protein, which means they don't get that normal flow of chloride ions. However, interestingly, heterozygotes who have 1 normal and 1 defective CFTR allele produce both functional and defective protein. Okay, so at the molecular level, the normal and the defective alleles are codominant because they both express because both alleles are expressed in the heterozygote. However, on the physiological level, because only 1 functional allele produces enough functional CFTR protein to allow that chloride ion for chloride ion transport, heterozygotes exhibit no adverse effects, and the mutated CFTR allele appears to be recessive at the physiological level. Okay, this is a type of dominance expressed by the allele that we can see in cystic fibrosis. Okay. Oops, let me go back. Another component in understanding the correspondence or the relationship between what is carried in the genes, the alleles, right, and then what is expressed phenotypically, goes back to these 2 concepts that I want to talk about, and that's penetrance and expressivity. Okay, penetrance and expressivity. Penetrance deals with the percentage of individuals having a particular genotype that express that expected phenotype. Let me give an example. Okay, if we were to look at those pea plants, right, big R, little r. We have assumed that in peas, the genotype big R, little r, which is heterozygote, always produces round seeds, and that the genotype little r, or homozygous recessive, always produces wrinkled seeds. However, for some characteristics, that assumption is not always correct. It's incorrect. The genotype does not always produce the expected phenotype. That phenomenon is called incomplete penetrance. Okay. So when the genotype does not always produce the expected phenotype, that is called incomplete penetrance. Okay. So another way of looking at penetrance, looking at this picture you see here of polydactyl. This is where humans have the condition of having the extra finger. Okay, there are several forms of it, but the trait is usually caused by a dominant allele. However, there are some occasions where people possess the allele for polydactyl, but have a normal number of fingers and toes. In that case, the gene for the polydactyl would not be fully penetrant. Okay, so how can we look at penetrance? Let's look at it from a numerical perspective. If it is the percentage of individual organisms having that particular genotype expressed phenotypically, let's look at the example of having 42 people that have the allele for polydactyl, and only 38 of them actually show polydactys. Okay, the penetrance would be about 90% or 0.9. Okay, how? 38 over 42. Now, something that is similar to penetrance, but not the same concept, is expressivity. Okay. Expressivity defines or looks at the degree to which the trait is expressed. So how much is the trait expressed? Okay. So similar to incomplete penetrance, where you have polydactyl that exhibit variable expressivity. Okay. Some individuals with this disorder possess extra fingers and toes and are fully functional, whereas others only have a small tag or like an extra piece of skin. Okay. So there are some instances where people can have incomplete penetrance and variable expressivity due to other effects like other genes being involved or environmental factors. What are some other factors that can affect the results of a genetic cross when you're looking at a single locus? Okay, because even the polydactyl is an example of that, but lethal alleles are another example. Okay, and a lethal allele is caused or causes death at an early stage, usually during the embryogenesis or embryonic development of the fetus. So before birth is actually occurs. Some genotypes do not appear among the progeny. We see this in mice, specifically true breeding yellow mice. And because of this lethal allele phenomenon, it affects the normal Mendelian genotypic and phenotypic ratios. For the example here in the yellow mice, you can see that there are no true breeding yellow mice. They found this, they did a study where they took yellow mice and they allowed them to breed, fertilization. And what they found was they had yellow mice and non-yellow mice. However, the number of offspring were not what they expected, and what they found was that there was the embryos, that were actually dead. They died in utero. They did not become fully or birth mice. And when they looked back at them, they were able to identify that they were homozygous dominant. That would be considered true breeding. So the only yellow mice that exist are carriers or heterozygous mice that are carrying the dominant trait. Because of that, the genotypic ratios are not normal because the true breeding yellow mice die in utero. There are many other genes that are associated with this, and this in part has to do with, and you'll hear some of this as you progress, is the Rally gene. There are certain genes that are associated with lethality. There are instances where you have the multiple alleles that also affect or deviate from the normal Mendelian principles and the ratios that we expect. There are some loci that have more than two alleles that are present within the group of organisms. And although there are more than two alleles, only two alleles will determine the genotype. But where multiple alleles are present for a given locus, it does alter the normal Mendelian principles. We see this in the ABO blood group, which we're going to talk about. And we also see this in the plumage patterns in ducks. The plumage patterns in ducks. So you can see here when you have the three alleles, you have the M, capital MR, lowercase md, and then M. I'm just right here. So 1, 2, and 3. So you have three alleles in this case here. The capital MR is restricted, which is more dominant. And then you have the capital M by itself, which is for mallid. And then you have M, lowercase m, and superscript D, which you see here. This is dusky. Also, just pay attention that the phenotypes of male and female mallid ducks do differ. But what we see is the restricted pattern of a female duck is shown for the parent. All the progeny 1 for the F1 generation have male plumage patterns. Have male plumage patterns. So another way of identifying this or looking at this is you have three alleles. But with the 3 alleles, you have 6 genotypes possible with this, with the 3 alleles. 6 genotypes possible. And you can look at this further on page 117 in the textbook. And it gives you the genotype with the associated phenotype. But we also have another example of the multiple alleles with ABO blood type, which is something that you all would be most familiar with. Okay. With ABO blood type, it refers to the antigens that are on the outside of our red blood cells. And these antigens determine our blood type, whether you are A blood, B blood, AB, or O. Now, these alleles encode for enzymes that convert the sugars on the outside of these red blood cells, these antigens that make them uniquely A versus B or AB, or lack thereof for O. Now, what is important about this is that these antigens can elicit an immune response when they encounter specific antibodies or complementing antibodies. If you have a blood type, okay, you have the genotype either homozygous dominant A, or you can be a carrier. Okay. The antigen type that you're going to present on the outside of your red blood cells is A, and the antibody that you're going to have in your body is anti-B. You should never produce antibodies against your own antigens, because if you do, that will be considered an autoimmune disease, and it would stimulate an immune response, which would not be good. Okay. It's like your body will be fighting itself. If you are B blood type, then you have these 2 types of genotypes, either homozygous dominant B or carrier for B. You are expressing the B antigen on the outside of the cells, and the antibodies that you are carrying are anti-A antibodies. For AB blood type, you have only one genotype, and that is because AB blood type exhibits codominance. The A allele is not dominant over the B, and the B is not dominant over the A, so they both exhibit or are expressed equally. But because they express both antigens, they don't express or present any antibodies. Because if they presented any antibodies, it would elicit an immune response against their self. Okay. Because of that, it makes the AB blood type the universal recipient. That means they can receive blood from anyone in the ABO blood family because they do not exhibit antibodies. Okay. O blood type is recessive. It only has this one genotype, which is little I. It does not express any antigens for AB. And so therefore, it's going to express both antibodies, A and B antibodies. So this makes O blood type the universal donor. It can donate blood to anyone, but it cannot receive blood from anyone. This becomes very important, one, because when you look at the blood type for ABO blood, it shows you that there's multiple alleles present. There's A, B, and I, okay, that produce different phenotypes, but it deviates from normal Mendelian principles in that manner in terms of the ratios. But most importantly, this is very helpful in understanding even blood transfusions. You have to know that your blood type, because you can elicit an immune response if you receive the wrong blood. Okay. But ultimately, hopefully, this helps you understand the genetics behind blood type and the actual blood transfusions. Again, more importantly, this shows a deviation from the normal Mendelian principles. So gene interactions. Gene interactions can take place when you have instances where genes are present at multiple loci, but they determine a single phenotype. Okay. With gene interactions, you have it affects genes at one locus, but it depends on the presence of genes at another locus. An example of that would be the interaction between genes that determine the fruit color of peppers. Okay. If genes exhibit independent assortment, or genes that, exhibit independent assortment, but do not act independently in their phenotypic expression. And then also products of genes at different loci that interact to produce new phenotypes. Let's look at this from the example of the peppers. So you'll see here in the P generation, the parental generation, you have homozygous dominant for the Y and C alleles or genes. And that produces a red fruit color. The cream will be produced phenotypically if there is a recessive allele for both. When they are crossed, the F1 generation are all going to produce red phenotype, and they are carriers. Okay. However, in the instance where the F1 are crossed with each other, the F2 show a 9-3 to 3-1 ratio. Okay. So if these are crossed, the F2 generation, after the crossing or inbreeding, will show a 9-3-3-1. That 9-3-3-1, or that dihybrid ratio, which deviates from the normal Mendelian principles, where you have the 3-3-1, however, it's still different in terms of the actual color. Okay. The F2 ratio in the first one produces one trait. But in the second instance, the 9-3-3-1 ratio in the dihybrid is 2 traits. But actually what you'll see here is we have 4 different phenotypes here. We have the red that is produced when both of the dominant alleles are present, Y and C. In the 3 instance here, you'll have a peach color when you have just one dominant Y allele, but the others are recessive. You'll produce orange, or sometimes what is referred to as yellow, when you have just one dominant C allele. And then you'll produce green, as you see here, when all are recessive. Not cream, I'm sorry. Not green, cream. Cream. So let's further look into how to solve this. And actually, before I go on, I want to just kind of show you all some math associated with this and looking at it from a Mendelian principles in terms of independent assortment with the single locus. Okay. So if we were to look at the case where we get red peppers here, this case right here, okay, we can look at the genotype. And what we see for the genotype, we have 1/2 from that's going to receive 1/2 will produce the dominant Y allele. And then for the dominant C allele, it's 1/2. These are independent events. So we apply the multiplication rule. 1/2 times 1/2. That's coming from it having one dominant allele and one dominant C allele. One dominant Y, one dominant C. That and the result overall gives a probability of 1 out of 4. Okay? 1 out of 4. And we could do the same thing for the rest of these. Okay? 1 out of 4. So what happens when you have these multiple instances for alleles at multiple loci and it produces a single phenotype? Okay? This whole concept is called epistasis. And this is where you have one gene that masks the effect of another gene. Okay. Recessive epistasis, and in each of these cases, it's going to deviate from normal Mendelian principles. Okay. It's going to deviate from normal Mendelian principles. And I'm going to give you some examples, but recessive epistasis is going to produce a phenotype of 9-3 to 4 in the F2 ratio. And dominant epistasis is going to produce a 12-3 to 1 in the F2 ratio. And duplicate recessive epistasis is a 9-7 ratio for F2. So let's look at it. Here in this case, we have the example of the ABO blood type. Okay. And this is showing the recessive epistasis. Okay. The recessive epistasis. The expression of the ABO antigen depends on the alleles at the H locus. Okay, because the H locus is the precursor to the antigens for A and B. They actually, the enzymes that are produced by the genes for A or B produce or convert the H allele to be A antigens or B antigens. So with that being said, Alleles at the ABO locus determine which type of terminal sugars are added to the compound. Okay. So here we'll see that for the ABO blood type, okay, specifically dealing with Bombay phenotype. Bombay phenotype is where they even have a recessive gene, both recessive alleles for the gene for the H antigen. And so as a result, they don't even have the H antigen. Okay, so because of that, the genotypes at both the H locus and the ABO locus determine the ABO blood type. And this is because when you look at the Bombay blood type, the phenotypes are recessive and they fail to convert that intermediate into the H antigen. Okay. However, the blood type O can result from the absence of the terminal sugar on the compound H. And so from the absence of the H compound, you get the Bombay blood type. But this goes back to the ability to mask, okay, the H antigen because of that defective enzyme. So in this case, in this example, the alleles at the ABO locus are hypostatic to the recessive H allele. They're hypostatic. Okay. What does hypostatic mean? Okay. Hypostatic refers to the alleles that are being masked. Okay. Hypostatic refers to the alleles that are actually being masked. And so in this case, the A, B are being masked because of the recessive mutation, the homozygous recessive mutation for the H antigen in Bombay blood type. Here is an example of yellow pigmentation in summer squash. And this is an example of duplicate recessive epistasis. Okay, dominant epistasis. And in this case here, you see that you have the compound A, right? The plants with this recessive WW produce enzyme 1. That enzyme 1 converts to the A to B to produce this green pigment. However, the dominant allele inhibits the conversion of the A to B, and so you don't get that color. Okay. Plants that have the Y produce enzyme 2, and that enzyme 2 converts the green to yellow. But if you have the plants with the genotype homozygous recessive Y, they don't encode that functional form of the enzyme, and so you don't get it. Okay. So ultimately, genotypes Y, W that are carriers, or the genotype with the W, the capital Y, or the dominant W, I'm sorry, with the lowercase recessive Y, don't produce the enzyme 1. And then the homozygous recessive for both produce enzyme 1, but not enzyme 2. And so ultimately it alters the normal Mendelian principles. And so what you see is a 12 to 3 to 1 ratio. In this example here with the snails, the pigment that is produced in the snails, this would be an example of duplicate recessive epistasis, and this is where you get a 9 to 7 ratio. This deals with the alleles, whether they're dominant or recessive, and their ability or lack thereof to produce the enzymes that convert the compounds for pigmentation. And in this case, because of the inability to produce the enzymes, it alters the phenotypic ratio. And in this case, the duplicate recessive epistasis produces a 9 to 7 ratio. A 9 to 7 ratio. Ultimately, what you end up seeing is 9 of the snails will have a pigmented shell and 7 will be albino. Okay, and that is because if you have any recessive alleles for A or the B gene, result in lack of pigmentation. Okay, so this is where you have duplicate recessive epistasis. Any of the recessive alleles present will produce a recessive phenotype, which in this case would be albinism. And here's a table that helps you to fully understand all of the ratios associated with various epistatic interactions, whether it's recessive, dominant, or duplicate. Okay. We're going to quickly go into complementation. Complementation is a term that helps you understand the determination to whether mutations are at the same locus or at a different loci. Okay, and a way to determine that is by using parents that are homozygous for different mutations, and you cross them. The offspring are going to be should be heterozygous. If the mutations are allelic, that means they occur at the same locus, they'll exhibit a mutant phenotype. If the mutations occur at a different loci, the heterozygotes from the offspring that inherit the mutant and the wild allele, they should exhibit a wild type phenotype. Okay, so this would be an example of complementation. And we see this concept in the complexity of the coat color in Labrador retrievers. Okay, and this has to do with what we call the agouti gene, okay, which is denoted by A, black, and E, and spotting. So it's very complex because you have multiple loci that are involved in producing a single phenotype, which is coat color. Okay, that can be, as you see here, the Labrador retriever, whether it's yellow, brown, or black, the beagle, or the Dalmatians, which it will be the spotting of the Dalmatian. Okay. And this table helps you identify and go into detail. I strongly suggest you guys go into this and understand this much better. I'm trying to make the lecture not so long so that you'll watch it, but make sure you go back and look at these this table. Lastly, we're going to go into the last two things: sex influence and sex limited characteristics. And you can see I've listed these examples for you and the figures associated with them. I really want to get to a couple of the diagrams to help further explain this. This is using goats and their ability to be beardless based off of sex, and sex influences how things are inherited. Sex influence characteristics are determined by autosomal genes, and they are inherited according to Mendelian principles, but they are expressed differently in males and females, and you'll see that here. This is a male goat that is beardless and a female goat, that is bearded. Okay, but pay attention to homozygous dominant versus homozygous recessive. They're allowed to mate, fertilize, and you'll see here in the F1 generation, the males are bearded. Okay, as long as they have one recessive allele, but the females are beardless if they have one dominant allele. Okay, so sex has an influence on the presence of the phenotypic expression. If you were to then take the carriers and breed them for an F2 generation, you're going to see that 3/4 of the males are going to be bearded. Pay attention to the genotype here, okay? And 1/4 of the females are going to be bearded. Pay attention to the phenotype, the genotype. This goes to show you that you can have that sex influences the inheritance and the expression of genes very, very much differently. And in part, it has to do with the higher penetrance in the sexes. Okay, it differs. We also see this in chickens, okay? Or in this case, a cock, okay? A cock is feathering in chickens. It's autosomal recessive trait and is limited to males, okay? So you see here, A is exhibiting the cock-feathered male, B is exhibiting the hen-feathered female, and C is the hen-feathered male. Okay, and you can also go back to see this example with puberty and as an autosomal dominant trait that is limited to males. I'm going to go past this one because I want to get to, where is it? This right here. I want to get to this component here, which was what we started off in the very beginning of the chapter. Okay, we'll end here. And that is sex influencing genotypes and the phenotypes. Now we see here that these are some snails, okay, and they were found to be, when they were discovered, that there was a small population that had a sinistral turn. Sinistral turn refers to a left-handed turn in the shell, versus the majority of these snails have a dextral turn, which is a right-handed turn. So what they did was Arthur Boycott went in and started to investigate this, and what he obtained 4 sinistral snails and began to study them and to breed them. Okay, and what he ended up finding was that shell coiling did not conform to Mendel's principles of heredity, and they eventually realized that dextral, the right-handed turn, was dominant to sinistral, but there was a twist to it. Okay, the phenomenon of a snail was determined not by its own genotype, but by the mother's genotype. Okay, this is termed the determination of the phenotype is called the genetic maternal effect. Okay, and this arises when the maternal produces a substance that is encoded by her own genotype, but is deposited into the cytoplasm of the egg that influences the development of that early offspring. So something from the mother's genotype is produced and put into the cytoplasm of the egg that is fertilized and then has an effect on the offspring. Okay. And this is what was found to determine the altered coiling in these snails to have a sinistral coiling or left-handed coiling versus a right-handed coiling. So it's called, another way of looking at it, the genetic maternal effect. The mother's genetic information determines or affects the supercoiling that one would experience here. All right, so at this point, I'm going to stop. And I want to strongly implore you all to finish going through the chapter and reading this, and go through the chapter summary and the visuals that are present. The practice problems are going to be very, very helpful for you. Okay.